for our module on lymphopoiesis 1. The lymphopoiesis is the process of formation of lymphocytes, which are white blood cells. There are groups of white blood cells that are separate from the rest, or are shown in the image below. They include the B and the T lymphocytes. And this particular module, we're talking about the process of formation of the B lymphocytes. Just an overview of the entire process of lymphopoiesis. It starts usually from a stem cell stage. The stem cell stage usually contains pluripotent cells that can undergo self-renewal and differentiation. Stem cells eventually differentiate and divide to form what we call committed cells or progenitor cells. And then progenitor cells would eventually differentiate and mature to form the precursor cells. Precursor cells are the cells that would eventually lead into the formation of mature cells. In the case of lymphopoiesis, we start with the stem cells. Stem cells become the progenitor cells. The progenitor cells usually tend to differ when talking about the B and the T lymphocytes, and then the progenitor cells would eventually become the precursor cells. Precursor cells will now go ahead to form mature cells. In the case of the T lymphocytes, we have the mature T lymphocytes, which could either be T4 or T8 cells. Alongside some other minor ones. And in the case of the B lymphocytes, the mature cells usually tend to become what we call the plasma cells. So diving into much detail, lymphopoiesis starts from the stem cell stage. Lymphocytes usually tend to be derived from the pluripotential hematopoietic stem cells, just like every other blood cell tends to be derived from. The pluripotential stem cells tend to differentiate into two major committed stem cells. This includes the myeloid stem cell and the lymphoid stem cell. In the case of lymphopoiesis, our focus is on the lymphoid stem cells and eventually what they give rise to. The lymphoid committed stem cells are cells that undergo self-renewal, cell differentiation, and in terms of a specific marker, they tend to be identified by the presence of the CD10 marker. CD10 marker, also known as cluster of differentiation, usually are receptors that are found on the membrane of cells. The particular subtype CD10 is what we call common all antigen. It's found in all forms of lymphocyte committed stem cells, either B cell or T cells. So this is not going to serve as a form of distinction between or differentiation between the stem cells that will become B cells and stem cells that become T cells. Usually, both of them would have the CD10 marker on their cell membrane. So the stem cells eventually divide and differentiate to form progenitor cells. The progenitor cell stage for this particular module, we're talking about the progenitor cells under the lineage of the B cell, and these cells are called the pre-B cell. In a separate module, we talk about the progenitor cells and the entire process of lymphopoiesis for the T cell lymphocytes. So talking about the pre-B cells are cells that are progenitor cells, hence they can't undergo self-renewal and they are not morphologically identifiable. However, they are committed to the formation of B cells. In terms of their location in the hematopoietic inductive microenvironment, they are found at the center, and we majorly use two major features in identifying pre-B cells within the bone marrow. The first is what we call immunoglobulin gene rearrangement. We'll talk about this in a separate slide. And then the second is the presence of cytoplasmic immunoglobulins. Remember that these are B cells, and the B cells are notorious for the production of antibodies, what we call immunoglobulins. These antibodies tend to already be found in the progenitor cell stage, usually within the cytoplasm of the pre-B cell. The pre-B cell eventually would undergo cell differentiation, division, and then mature to form the precursor cell stage known as the B cell in transition. We'll talk about that when we get to that aspect of the model. So let's focus a bit about the concept of gene rearrangement. In a pre-B cell, the gene for formation of antibodies has the capacity to rearrange, and this leads to the increase in variety of antibodies that can be produced. However, this leads to what we call loss of germline inheritance, and I'll explain what this means. So think about a B cell. The purpose of a B cell is the production of antibodies or immunoglobulins. For antibodies to be formed, usually would require the process of the central dogma of life, which is process of replication, transcription, and translation of mRNA into protein, and the proteins would eventually give rise to the antibodies. The genes for this particular central dogma of life is already present within a particular pre cell. So a pre cell would have what we call a germline inheritance, in which a particular type of gene for a particular type of antibody is already inherited um, from the parents of that particular individual, hence the reason why it's called the germline inheritance. But during the process of the progenitor cell stage of lymphopoiesis, the gene that was inherited from the parents tends to undergo rearrangement, and that rearrangement would give rise to different forms of antibodies being formed. So let's imagine that we had a particular form of antibody, a particular form of gene that gives rise to a particular form of antibody. If that gene becomes rearranged, a different form of antibody will be produced through the central dogma of life, and that's what we mean by gene rearrangement. So usually, the pre cells in the progenitor cell stage of lymphopoiesis has the capacity to undergo gene rearrangement, and as a result tends to undergo loss of germline inheritance. These are two important concepts to remember. So to elaborate more on why this is extremely essential to the process of formation of antibodies, an antibody tends to contain two major chains, heavy chain and then the light chain. The heavy chain is shown in brown, and then the light chain is shown in blue. And then each of these chains tend to have different fragments. So the heavy chain, for example, has four major fragments. This includes the variable region. The variable region is what this area marked here. So this is the variable region. Then we have the diversity region. Diversity region is down below. Then we have the join. This is diversity region. And we have the joining region at this point. And then finally, we have the constant region down here. So these are the four major regions of the heavy chain. And these are composed of different forms of antibodies. And as a result, they are gotten from different aspects of genes in the body. So we have genes that will code for the variable region. We have genes coding for the diversity region. We have genes coding for the constant region, and we have genes coding for the joining region. Each of these genes are numerous and usually and first of all inherited by the parent of the individual. That's what we call germline inheritance. And as a result of lymphopoiesis, these genes could rearrange, and once they rearrange, the form of antibody or protein that they eventually produce through the central dogma of life would differ, and then the combination of these proteins will lead to the combination of a different form of heavy chain. The light chain also tends to vary in its proportion. The light chain is what is shown in blue. We have two major subregions of the light chain. The light chain has a variable region in V and then the constant region in C. And the different forms of combination and permutation that we have for different forms of genes in either the heavy chain or light chain could result in varieties of antibodies being formed once gene rearrangement occurs. I'll explain this in elaborate terms in the next slide. So this is a slide that explains for us the process of gene rearrangement and eventually the production of varieties of antibodies. So you can see a lymphoid stem cell undergoing maturation into an immature B cell through the process of lymphopoiesis. You'll notice that the heavy chains and light chains of the antibodies are already being formed from the genes. So these are the genes present within the individual. What happens is that for the heavy chain, we have four major types of regions on the heavy chain, and each of those regions have different forms of genes. So for example, the variable region or the heavy chain has 40 different genes that can form it. The diversity region has about 10 different genes that could form it. The joining region has about 6 different genes that could give rise to it, and then the constant region has about 9 different genes that could give rise to it. And each of these genes could be picked at any point in time to form a particular set of antibody, and then the antibodies could be combined in different combination or different permutation to give rise to the heavy chain. The light chain, on the other hand, also has different forms of genes that gives rise to its variable and constant region. For the variable region of the light chain, we have about 40 genes that could give rise to it, and then the constant region has about 3 genes. So just imagine the amount or level of combination and permutation that we could have when we have different genes combining together to give rise to the heavy chain and then the light chain, and then the combination of two of them will give rise to an antibody. So what happens is that first of all, when a particular stem cell or is produced and the stem cell becomes a progenitor cell, the initial baseline gene, which was the germline inheritance, becomes rearranged, and that rearrangement leads to varieties of immunoglobulins being formed, not just the particular type of antibody that that cell could produce that was gotten from the um, parents, gotten from the germline inheritance, but because of rearrangement, you have varieties being produced as a result, and that's what the germline rearrangement or gene rearrangement tends to entail when it comes to the process of lymphopoiesis. This is very key when talking about the pre-B cell, particularly the progenitor cell of the B lineage of lymphopoiesis. Let's move on to the precursor stage. So once we have the pre-B cell undergoing maturation and differentiation, it gives rise to the first precursor cell in the B cell lineage of lymphopoiesis. This is known as B cell in transition. The distinction between the B cell in transition and the pre-B cell is the presence of both a cytoplasmic and a surface level surface membrane immunoglobulin. Remember that for the pre-B cell, there are two major things that we use in identifying them: the presence of gene rearrangement and the presence of cytoplasmic immunoglobulins. For the B cell in transition, we have both surface membrane immunoglobulins. So we have immunoglobulins on the cell membrane, and we have immunoglobulins present within the cytoplasm. Also, alongside the CD10, we also have some other clusters of differentiation or receptors found on the cell membrane of the B cell in transition. This includes CD19, CD20, CD21, and 22. In fact, when we see CD20, 19, 21, we are thinking about the B cell lineage of lymphocytes. B cell usually would undergo cell maturation and differentiation and give rise to the next precursor cell known as the virgin B cell. The virgin B cells are also called circulating B cells. They are cells that have migrated out of the bone marrow and into the bloodstream, and hence the reason why we call them circulating B cell. You might be wondering why we call them virgin B cell, because this is because of the fact that the virgin B cells are cells that have not been antigenically stimulated. They have not been exposed to antigens, and as a result, they are fresh cells that have not been disvirgins, quote unquote. They, in terms of their structure or in terms of their identification, they possess only surface membrane immunoglobulins, and this is the um, distinct from the B cell in transition that contains a combination of surface and cytoplasmic membrane immunoglobulin. So the antibodies for virgin B cells are found only on the membrane and not in the cytoplasm. They also have clusters of differentiation 19, 20, 21, and 22. And when they become antigenically stimulated is when they undergo further maturation. Usually, the virgin B cells become activated or become antigenically stimulated when they come in contact with a corresponding antigen. So remember that they have a surface membrane immunoglobulin, they have a surface antibody. So let's imagine that the surface antibody on the virgin B cell comes in contact with an antigen. Let's say it is an antigen. Once that contact is made between the antigen and the surface membrane immunoglobulin, that would lead to antigenic stimulation. This leads to the virgin B cell expressing CD23, and then that will lead to the virgin B cell now migrating to the lymph node. So they move from the bloodstream into the lymph node, and at that lymph node they start to mature. The very first cell that they mature into is what we call the cleaved B cell, and this is the next step of the process of lymphopoiesis for the B cell lineage. So the cleaved B cells are B cells or precursor cells of the B cell lineage that are found in the lymph nodes. They are derived from the virgin B cells after antigenic stimulation has occurred. They are cells that only contain surface membrane immunoglobulin, just like the virgin B cells. And in terms of the clusters of differentiation, they have CD19, CD20, 21, 22, and then the newly acquired 23, which occurs during antigenic stimulation. Cleaved B cells are found particularly at a site in the lymph node. We need to remember this is called the follicular center of the lymph node. If you look at the picture down below, you notice that the lymph node has different areas. It has the parafollicular region, it has the medulla, it has the cortex, it has the germinal center. But if you pay attention, the smaller circle present here is the follicular area. The inner aspect of that follicular area is called the germinal center. That's where we tend to see most of the mature B cells. But B cells that are undergoing maturation, like the cleaved B cells, will be found in the follicular region. That's just the outer coating, that dark blue area. So the cleave cells usually tend to exist in two stages. They exist in small cleave cell stage and the large cleave cell stage. Usually we start with the small cleave cell, and then this would mature into the large cleave cell. And then from there, the large cleaved B cells would undergo cell division and differentiation to form the non-cleaved B cell. And that's the next stage under the precursor cell stage of the B cell lymphopoiesis. The non-cleaved B cells are also found in the follicular center. They also possess the surface membrane immunoglobulin and also possess CD19, 20, 21, 22, and 23 that we got from antigenic stimulation. And then the non-cleaved B cell also has two major stages. It has a small non-cleaved stage and a large non-cleaved stage. Then it undergoes further maturation to give rise to the next process or next stage under lymphopoiesis, which is what we call the B immunoblast. So B immunoblasts are unlike the other cells, they are found particularly in the bone marrow. So these are cells that migrate from the lymph node into the bone back into the bone marrow. And once they get into the bone marrow, they tend to undergo further maturation to form the plasmacytoid cells or the memory B cells. So first of all, the B immunoblasts have very similar characteristic to that of the non-cleaved and cleaved B cells. So they contain surface membrane immunoglobulins on their membrane. They have CDs 19, 20, 21, 22, and 23, just like the cleaved and non-cleaved B cell. But they could either undergo two forms of differentiation. They could differentiate further to form plasmacytocytes. We talk about that in the next slide. Or they could revert back to a morphological appearance that is similar to the virgin B cell. And in this case, although they are not practically reverting to virgin B cells, they just revert back to cells that appear like the virgin B cells. These cells are called memory cells. We'll talk about the memory cells next. So the memory cells are virgin-like B cells. They are cells that appear like virgin B cells, but they've already undergone antigenic stimulation. That's the difference. So memory B cells are cells that appear like virgin B cells, but have undergone antigenic stimulation, have gone through the lymph nodes, come out of the lymph nodes, and gone to the bone marrow. So cells that have become cells that were coming from virgin B cells to cleave cells to non-cleave cells to be immunoblasts, but revert back to memory B cells and appear like virgin cells. These are cells that become memory B cells and have undergone antigenic stimulation. What is unique about the cell is their capacity to undergo maturation directly back into B immunoblasts. So whenever the cells are re-exposed to antigens, they undergo another set of antigenic stimulation instead of going through the entire process of becoming cleave cells, non-cleave cells, and B immunoblasts. They just go from memory B cells to B immunoblast straight up. Um, the unique thing about them, that's the unique thing about them. So besides that, they also possess surface membrane immunoglobulins and they contain the amount of clusters of differentiation that we saw in the B immunoblast and the non-cleaved and cleaved B cells. The second form of precursor cells that the B immunoblasts could mature into are the plasmacytocytes. They are also found in the bone marrow. They possess surface membrane immunoglobulins, shown in the image. They express CD19, 20, 21, 22, and 23. And these are the cells that lead directly into the complete maturation of B cells. And the plasmacytocytes will differentiate to form the plasma cells. And these are cells that are able to secrete the surface membrane immunoglobulins to antibodies into the bloodstream. So at the end of the process of lymphopoiesis for the B cell lineage, we have a single virgin B cell undergoing multiple cell differentiation and division to give rise to a total of about 128 plasma cells. Plasma cells, remember, are cells um, that are large in size, possess CD10, CD19, 20, 21, 22, 23, and they have the special ability to secrete surface immunoglobulins as antibodies into the bloodstream. The entire process of lymphopoiesis in terms of the B cell lineage tends to take about 2 weeks, about 14 days. So these 14 days would encompass from the stem cell stage as the pre-B cell all the way to the plasma cell as shown in the image. The lymphopoiesis as a whole is regulated by a number of different factors. So we have the granulocyte colony stimulating factor, stem cell factors, and interleukin 3 being the major factors involved in the process of driving lymphopoiesis to its conclusion. What are the various pathologies that could have in the process of lymphopoiesis? In the process or in the case of an increased production of lymphocytes, we call that lymphocytosis. So this would either be a B lymphocyte or a T lymphocyte, depending on the scenario. Usually, B lymphocytes tend to increase when there's a bacterial infection. T lymphocytes, depending on the type, could increase whether there's a bacterial or viral infection or a tumor being present. When there's a decreased production of lymphocytes, this should lead to a process called lymphocytopenia. Malignancy of the pre-B cell and the B cell in transition usually causes what we call an acute lymphocytic leukemia. This is a form of blood cancer. Then a malignancy of the virgin B cell usually leads to a chronic lymphocytic leukemia. When we talk about malignancies of the cells found within the lymph nodes, that's the cleaved B cell and the non-cleaved B cells, usually that will lead to a form of malignancy we call lymphoma. And then finally, malignancies of the B immunoblasts in the bone marrow, the plasmacytoid or the plasma cells themselves, usually leads to a form of malignancy we call multiple myeloma. And that concludes our module on lymphopoiesis 1.